I'd like to introduce my very good friend and colleague, Dr. Dan Kastner, who obtained his AB, summa cum laude, in philosophy from Princeton University in 1973, and a PhD and MD from Baylor College of Medicine in 1982. After completing internal medicine residency and chief residency at Baylor, Dan moved to the National Institute of Health in the residency in 1985, and he's currently an NIH Distinguished Investigator, Chief of the Inflammatory Disease Sections, and Scientific Director Emeritus of the Division of Intramural Program at the National Human Genome Research Institute. That emeritus is very important to me because Dan trained me, and I'm not doing his function as the scientific director. Again, opportunity to thank Dan for making the house very stable for me to step in. So thanks, Dan. Throughout his career at the NIH, Dan research has focused on using genetic and genomic strategies to understand inherited disorders of inflammation, often stimulated by patients with relatively rare disorders seen at the NIH Clinical Center Hospital. This work has provided detailed molecular explanation for these illnesses and has established the conceptual basis for highly effective targeted therapies, and has informed our understanding of more common illnesses. Dan's group also proposed the now widely accepted overarching concept of autoinflammatory diseases to denote disorders of the evolutionary ancient innate branch of the human immune system. Dan has won a number of awards and honors. That is absolutely correct, and the list will go on, but I'm just going to summarize it quickly, including election to the National Academy of Science in 2010, to the National Academy of Medicine in 2012, and recognition as the Federal Employee of the Year in 2018, the Rose Prize in Molecular Medicine in 2019, and the Crawford Prize in polyarthritis from the Royal Swedish Academy of Science and the Crawford Foundation in 2021. So I welcome Dr. Kastner. Thank you.
Well, Charles, thank you so much for that very kind introduction. And I want to thank you and all of the Board of Scientific Directors for having chosen me for this very high honor of giving this the 2023 G. Burroughs Mider Lecture. And we have Dr. Mider on the slide right here, the opening slide. He was the first NIH Director of Laboratories and Clinics, which I think is the equivalent of the Deputy Director for Intramural Research. And I hope that during the course of my talk that you'll see how we can really bring together what goes on in the laboratory and what goes on in the clinics. In this particular case, looking at autoinflammatory disease and the human condition. But before we launch into that, I must remind you that I have no financial relationships to disclose. So in any event, starting out, maybe for those who are not familiar with the systemic autoinflammatory diseases, if there are any such people among you, these are a group of disorders in which patients have recurring episodes of seemingly unprovoked, localized, and/or systemic inflammation without the high-titer autoantibodies or antigen-specific T cells that one would typically see in the autoimmune diseases, or evidence of overt infection. And these patients can have dramatic evidence of inflammation. I'll just illustrate with a few photos here on the slide. This happens to be the forearm of a young man from Kansas City with Fapa syndrome, the syndrome of pyogenic arthritis with pyoderma, gangrenosum, and acne. This is, in this particular case, the skin lesions, one of the skin lesions that these patients can have. This took several months for this patient to heal. Here we have an MRI of the leg of a patient with TRAPS, the TNF receptor-associated periodic syndrome. These patients can have inflammation going down into the muscle compartment, a fasciitis that migrates down the leg. Next, a relatively new autoinflammatory disease, ROSA, that oftentimes presents with vision loss, but it is a systemic disease, and in fact, these patients also develop, as you can see, a polyarticular arthritis. And then finally, our last example is DADA2, the deficiency of adenosine deaminase type 2, which is a disease in which young children can present with unexplained fevers, sometimes strokes, or sometimes inflammation of the blood vessels, of vasculitis. So, in any event, these turn out to be, as Charles said in the introduction, disorders of the phylogenetically ancient innate branch of the immune system, and they have provided new insights into human biology and opportunities for targeted therapies. Now, perhaps to put this in a little bit of historical perspective, I project the image of Paul Ehrlich. Paul Ehrlich was one of the great immunologists of the early 20th century. He won the Nobel Prize in Physiology or Medicine back in 1908, and he proposed the concept of horror autotoxicus. That concept was that the immune system is so potent that it would be impossible for it to turn against its host. And therefore, the central dogma, or at least a dogma, of immunology in the early 20th century was that autoimmunity was impossible. In about 1950, Ivan Reut discovered autoantibodies against the thyroid in patients with Hashimoto's disease, and shortly thereafter, a number of other autoantibodies were discovered in other autoimmune diseases. And so the latter half of the 20th century then was the time when autoimmune diseases of the sort that we usually think of were first characterized. But it was not until the end of the 20th century, actually, that this concept of autoinflammatory disease came to the fore. Now, my first exposure to a patient who turned out to have what we now call autoinflammatory disease was actually when I was a beginning fellow in rheumatology here at the NIH in the Clinical Center. Here you can see this was a handwritten note that I wrote on November 14th of 1985 after I had seen this patient in clinic, a young man of Armenian ancestry who had a lifelong history of recurrent fevers and mono or pauciarticular arthritis. He turned out to have familial Mediterranean fever, which is sort of the prototype autoinflammatory disease. For those of you who aren't familiar with FMF, it's a disease that at the time I first saw that patient in 1985, it was recognized to be an inherited disease, a recessively inherited disease, but no one knew what the gene was or anything about the pathophysiology of the disease in any serious way. In any case, it's seen in people of Armenian, Arab, Turkish, Italian, and Jewish ancestry, and it's characterized by relatively short, on the order of 1 to 3 day episodes of fever with sterile peritonitis or sterile inflammation of the lining around the lungs. Some of the patients develop arthritis or arthralgia, and at least in some cases, patients will have the buildup of a cleavage product of one of the acute phase proteins, one of the inflammatory proteins that deposit in the kidneys and can lead to kidney failure and death. So this was definitely a very interesting disease, a very dramatic disease for me, seeing the patient. There was actually a treatment, colchicine, for this patient, which was great. But it did sort of get me interested in that disease. And this was right at the time that was this magical time at the beginning of the Genome Project, when all of a sudden one had at least the conceptual possibility of doing at scale some sort of a screening of the whole genome in an unbiased way to discover the cause of a disease that you didn't have a clue as to what was going on. So that was really fantastic, and eventually, it took a little bit of time for me to get involved in this, but by the summer of 1989, I was hard at it in terms of trying to find the gene for familial Mediterranean fever using the new tools of the Human Genome Project. However, because of the fact that there were very few markers in the human genome that were known at that time, only about 300 markers, DNA markers in the whole human genome, you had to have lots of families in order to be able to actually map a disease gene. And so here you see the expedition that I went on in the summer of 1989. And here I'm visiting a family in Akko, a coastal city in northern Israel. You can see the photo has been modified to meet HIPAA requirements so that we're not identifying anyone. And you can see my face there, with most of the images there. This is Professor Mordecai Pross, who was my collaborator in these adventures. And in any case, this particular family is a family where the parents were first cousins of one another, and you can see the familial resemblance between the two of them. And in any event, but then they had several kids that actually had FMF, so it was a great family for doing the genetic linkage analysis. Altogether, we drew blood on 347 individuals and made EBV cell lines on these people. That's what you did back in those days in order to be able to map a disease gene. And after a couple of years, we were successful in mapping the gene to the short arm of chromosome 16. And then we became the Genome Project for that area of chromosome 16, developing genetic maps and physical maps and transcriptional maps, and then sequencing all of the genes. We discovered 10 genes in a 200 kb interval that then we had to screen for mutations. And ultimately, it was the 10th of those 10 genes that turned out to have the mutations that actually were associated with FMF. And we found just what we had been hoping for, this brand new gene that nobody knew anything about it before, and we named the encoded protein pyrin after pyrexia for fever. And so that was really exciting. But then, of course, it became clear to us that actually we didn't know anything about how this protein worked or what it did or whatever. And so then, we were actually faced with the puzzle of figuring out how that all worked, which has been something that's taken us a little bit of time to figure out. But one of the early clues, anyway, is that the N-terminal 90 amino acids of this protein is the prototype for a domain, which is known as the pyrin domain. And it forms 6 alpha helices. You can see them here. And that allows for the formation of a charge dipole with positive charges in blue, negative charges in red, and basically that allows for the self-self interaction, the cognate interaction of the pyrin domain from one protein with the pyrin domain from another protein. And that's actually how a number of the proteins in the innate immune system interact with one another. And there are some 20 different proteins in the innate immune system that have pyrin domains. So that was one of the great things that we learned early on. And then the other thing that was really great too, and this is where, keeping that connection between the lab and the clinic is so important, is that we started seeing patients that came to the NIH who were referred with the possibility that they might have FMF. And we had a genetic test, so we could tell, if they did or not. And some of them did, but then there were some of them that did not. And we actually identified a cohort of individuals with dominantly inherited, rather than recessively inherited, recurrent fevers, the attacks lasting a long, long time, having a different ethnic distribution and a different response to treatment from FMF. And actually, it then turned out that Ivona Aksentievich and Mike McDermott, a couple years after we found the gene for FMF, found that those patients had mutations in the P55 TNF receptor, the receptor for tumor necrosis factor. So we called that disease TRAPS, the TNF receptor-associated periodic syndrome. But we thought that maybe, just maybe, we were seeing an emerging family of new diseases that were different from the autoimmune diseases because these patients didn't have autoantibodies or any evidence of T-cell involvement. And so we thought to call this family of diseases the autoinflammatory diseases instead. But it was just, sort of a guess, that might be the case. The following year, actually, we published an opinion article in which we speculated on all of the different diseases that there might be that could be autoinflammatory just based on the idea that these patients had inflammation without autoantibodies. I mean, that was it. That was how we came up with this list. And we had this classification scheme, and you can see that a couple of the diseases fit into this familial urticarial syndromes, in which you have the familial cold urticaria, where patients, if they are exposed to cold, they develop fever and break out in hives, or Muckle-Wells syndrome, where they don't need cold to be induced to do that. So that was our speculation. And then it was magical what happened. So what happened was within about a year, Hal Hoffman at the University of California, San Diego, published this article in which he identified the gene that's mutated in those two diseases, those self-same diseases that I just showed you on the last slide. And it turned out that the gene that is mutated in those diseases has at its N-terminus a pyrin domain. So that was like, wow, so there actually is a connection amongst these diseases. So that was great, just for us. But then the other thing that was happening was that in Switzerland, there was this guy named Jörg Champ, and he was interested in how IL-1 gets activated. There's a pro-form of IL-1, and it has to get cleaved in order to be activated, and he figured out that there was this macromolecular complex called the inflammasome, and guess what was actually at the hub of that macromolecular complex? NLRP3, which is the same protein that actually is the protein that Hal Hoffman found with the pyrin domain at its N-terminus. So all of a sudden, we had some vindication of this idea of autoinflammation, but also a clue that these diseases had something to do with IL-1. And then, as if this story is not miraculous enough, there's another piece of it too, and that is that Ivona Aksentievich and Rafaela Golbach-Mansky discovered that an even more severe disease than the ones that I just told you about, in fact, also have mutations in that same gene, and that's a disease that's called NOMID, neonatal onset multisystem inflammatory disease. And those patients not only get fevers and hives, but they also develop aseptic meningitis, which can lead to blindness and deafness and intellectual disability. So it didn't take, and this is just a diagram showing that the mutations are all in the same domain for those three diseases. So in any case, that led the good Dr. Golbach-Mansky to organize a trial here at the NIH Clinical Center looking at an IL-1 inhibitor in 18 children that have NOMID, this neonatal onset multisystem inflammatory disease. And this is a disease that the prognosis in these kids would be horrible. And if you look at the before and after of what happened, so before treatment, you can see the hives like rash, conjunctivitis. This is an MRI of the brain. The white indicates inflammation of the meninges. And then the arrow here points to inflammation of the cochlea. And then, after being on this injectable inhibitor of IL-1 called anakinra, what you can see is that the hives like rash goes away, the conjunctivitis goes away. That happens within a day. And then within months, the aseptic meningitis goes away and the cochleitis goes away. And so you had a cohort of children who had a horrible prognosis who now are still on anakinra. We saw one of them at a meeting on autoinflammatory diseases about 3 weeks ago. He's now a graduate student studying autoinflammatory diseases. So it's just really a wonderful, wonderful outcome of that. So in any case, the field has taken off at this point. Mike McDermott published a paper just showing that, or proposing that autoinflammatory diseases and autoimmune diseases are sort of two ends of a spectrum, which is probably the case. Seth Masters, when he was in my lab, published an article that sort of summarized 10 years after the proposal of the concept of autoinflammation, what was the molecular classification, at least at that time. And sort of tongue-in-cheek, he came up with this title, Horror Autoinflammaticus. So remember that Ehrlich's concept, horror autotoxicus, was that autoimmunity, autoinflammation was impossible. Well, this title, basically is introducing an article where there's all sorts of autoinflammatory diseases that are being described and then discussed in terms of their pathophysiology. Shortly thereafter, Phil Hashkus, who's here actually in the auditorium, and Ron Laxer and Anna Simon published a textbook of autoinflammation, 800 pages or something like that long. And just recently, there was a meeting of the International Society on Systemic Autoinflammatory Diseases with something like 360 people there, and more than 50 monogenic autoinflammatory diseases now known. So it's not going to be possible in the 5 hours that has been allotted to this lecture for me to go through all of the 50 diseases that we could talk about. So I'll talk about one of them that is important because of its frequency, because of its severity, and because there's a treatment for it. And then another one that's a real recent one. So the first one is this disease, DADA2, deficiency of ADA2. It's a disease that we discovered, and actually Phil Hashkus and his colleagues discovered independently at about the same time, and the two publications were published back-to-back in the New England Journal. We had seen a number of kids here at the NIH Clinical Center that presented with fevers and recurrent strokes. Phil had seen a group of children with fevers and sometimes inflammation of the blood vessels, polyarteritis nodosa. But they both turned out to have mutations in the same gene, which is this gene ADA2, adenosine deaminase type 2. You can see here on this slide, the arrows are pointing to strokes in the deep brain nuclei. These kids can have a livido kind of skin rash. This is vasculitis, a necrotic area of the ear in one patient with polyarteritis nodosa. In any case, a problem with this was that these kids continued to have strokes no matter what the treatment seemed to be. And shown here is Dr. Amanda Umbrello, who published a paper about 5 years after we had discovered the disease, showing that TNF inhibitors are very, very effective in preventing strokes in these patients, and that's become standard of care at this point. The other disease that I want to mention is disabling pansclerotic morphia. Now, this is a disease that's not a household name, I don't think, or at least not in most households. And it's a disease in which patients have thickening of the skin, a form of fibrosis that goes down deep below the skin, subcutaneous tissue, muscle tissue, all the way to the bone. These patients also can have problems with poor wound healing, and they can develop squamous cell carcinomas. And the prognosis for these patients after diagnosis is maybe to live about 10 years. So it's a very severe disease. So it just happened that I was giving a virtual grand rounds to the University of California, San Diego about a year ago. It was February of 2022. And at the end of my talk, I was meeting with various people virtually. Everything is imaginary, whatever. So in any event, I was talking with Lori Broderick, who's on the faculty there. And she was telling me about this family that she cared for with this disease, disabling pansclerotic morphia. And so the story was horrific. It was the story of a young man who was about 20 or so, who had this condition. And it had progressed to the point where he had had a quadruple amputation, where both arms and both legs had to be amputated because of uncontrolled infections and squamous cell carcinomas. And then, as if that wasn't terrible enough, his older brother started to develop the same kinds of lesions. And so at that point, the physicians caring for that family referred the family to Dr. Broderick, who did exome sequencing and found a gain of function, what looked to be a gain of function mutation in STAT4. But the problem was that she only had the one family, and so she couldn't really publish this, because usually you have to have a confirmatory family. Now, it just so happened that our group, collaborating with Cassie Torak at the University of Pittsburgh, had the missing link in this puzzle. We had another family where there was a child with a de novo mutation that also had a similar phenotype. And then actually, Bill Gall in the Undiagnosed Diseases program had a third family. So all of a sudden, instead of it being just a singleton case, it became a more convincing story. John O'Shea was also important in helping us through all of the signaling vicissitudes with STAT4, and his excellent postdoc, Rachel Phillips, and the outstanding MD PhD student, Sarah Blackstone, in my lab did a lot of the functional studies. So just a little bit about this. Here's just some photos of patients with disabling pansclerotic morphia. Here are the pedigrees of the families, our de novo family over on the right, and then the two other families, Laurie Broderick's on the left and the Undiagnosed Diseases Program family. And then the mutations, they're all in the SH2 domain, which is an important activation domain of STAT4. So in any case, one of the first things that we needed to do was to actually demonstrate that these are gain-of-function mutations. And this image just shows that comparing STAT4 phosphorylation between the wild type, which is in blue, and the other colors, you can see that the other colors are shifted to the right, and there's increased phosphorylation plotted in this way. And then if the cells are stimulated, that was unstimulated, if the cells are stimulated, there's persistent phosphorylation compared with the wild type. Laurie Broderick also did an assay looking at wound healing in vitro. And there, if you take a lawn of fibroblasts and scratch it with a pipette tip, then what should happen is that over time, the fibroblasts will grow back together, and you can measure how long that takes. And you can see here that for the wild type, they close up, the cut closes up within about 24 hours. For the mutation, it's still open at 24 hours. And if you look at the supernatants of those cultures, there's a lot of IL-6 being released into the supernatant with the mutation. And in fact, if you look at the wound healing, if you take either comparing, comparing the wild type with the mutant, you can see the mutant doesn't close up. But if you add IL-6 to the wild type, it gets also having a problem with regard to wound healing. So it appears that IL-6 is playing at least some role in that. This suggested a possible positive feedback loop because you see IL-6 is downstream of STAT4. So IL-6 can be produced. The fibroblasts have receptors for IL-6, which then signal through STAT4. So, the Janus kinases are involved in that process. So using a JAK inhibitor then would make a certain amount of sense. And one can see that in vitro, using ruxolitinib actually does reduce the IL-6 that's being produced in vitro. One can see that in terms of the wound healing assay, that in fact one can normalize the poor wound healing of the mutant cells with ruxolitinib. And then if you look at the before and after with the patients, in fact, the older brother of that family that I said was beginning to develop the same kind of lesions as his younger brother actually responded quite well to ruxolitinib. So this is hot off the presses. In fact, it was published in the New England Journal of Medicine online exactly 1 week ago today. So it hasn't even gotten into the print version of the journal yet. Well, in any case, the title of this talk, it's a very high-sounding title, Autoinflammatory Disease and the Human Condition. And I haven't said, I have said a lot about the condition of some humans, but what in general can we say about autoinflammatory disease and the human condition? I'm sure that either you've forgotten that question by now or you're just dying to know. So in any case, the first thing that I can say is that we've learned that microbial infection, innate immunity, and cell biology are intrinsically related. And one of the things that has come out of the study of these diseases is that we've slowly but surely figured out the mechanism of the disease, more than just knowing that such and such a gene is mutated. So I'll just show you this one example of FMF. And it actually took almost 20 years from the time that we found the gene to the time that we figured out this pathway. And a number of other people were involved in this too. So in any event, the story actually, you might think, that if pyrin, the FMF protein, is some sort of a sensor for something that's a receptor for some sort of microbial pathogen, that you're going to have some sort of a diagram where some microbial pathogen binds to pyrin. And that was sort of the mindset that we had at the beginning, is that that would be the way it would work. But it turned out that that was wrong. So in this figure, you can see just here that these are two pyrin molecules that are beginning to form an inflammasome. But the regulation of it actually occurs, it starts here inside the cell membrane with a molecule called Rho-A. Rho-A is a GTPase that can activate certain kinases, in this particular case PKN1 and PKN2, which lead to the phosphorylation of pyrin, and then phosphorylated pyrin is bound by these inhibitory 14-3-3 molecules. And so in the normal situation where you have all of this working, you have basically a tonic inhibition of the pyrin inflammasome. But there are certain bacterial toxins that actually will disrupt Rho-A. And when that happens, then you don't get phosphorylation of pyrin, you don't get the binding of the inhibitory protein, and you can get activation of the pyrin inflammasome and release of IL-1. So then, the question is, well, why exactly would that happen, that Rho-A would get inactivated? And it turns out that certain bacteria make toxins that inactivate Rho-A. And they inactivate Rho-A probably because Rho-A is actually a key molecule for assembling the actin cytoskeleton. And the actin cytoskeleton is really, really important for a lot of white blood cell function. It's important for phagocytosis, it's important for migration, it's important for degranulation. And so white cells need to have a functioning Rho-A. Bacteria then have evolved toxins that inactivate Rho-A as a way of basically attacking the host. And the pyrin inflammasome is the evolutionary response to that. And, so this is an example of where the innate immune system works indirectly. And in fact, this is oftentimes what's happening, is that the innate immune system is sensing changes in cellular homeostasis rather than that it is actually directly binding to a bacterial or viral product. So it's a very interesting thing with regard to how things work. This is just a photo of a patient that has a mutation in the phosphorylation site of pyrin. And so under those circumstances, those patients have continuous inflammation. Rather than a periodic fever syndrome, they're inflamed all the time, and they have a neutrophilic dermatosis. What's more, just to follow up on this story with bacteria, we have sort of this cat and mouse relationship with various bacteria. And so here's the story of Yersinia pestis, the agent that causes bubonic plague. And so Yersinia pestis makes a couple of toxins, YAP-E and YAP-T, that activate the pyrin inflammasome. And so you would say, okay, so the pyrin inflammasome is a defense against plague, and that, at least on the surface, would be right, except that plague has evolved another toxin, YAP-M, that actually can phosphorylate pyrin. It actually hijacks another kinase in the cell to induce it to phosphorylate pyrin. And so that blocks the pyrin inflammasome. And so you have this back and forth between Yersinia and humans. And then the next move is actually in FMF. And so you see the increased phosphorylation of pyrin by YAP-M in healthy individuals on the left. But in individuals who have FMF, they actually don't see increased phosphorylation of their pyrin. So FMF, the carrier state for FMF is actually a defense against plague. And so one would think then that perhaps this leads to selection for FMF mutations. And so we looked in the Turkish population to see if there was any real evidence for selection of FMF mutations. And so one of the telltale signs of selection is if you have a very big haplotype, a big DNA fingerprint that's associated with the mutation. And you can see the width of this blue peak here. That's the width of haplotypes that are associated with the FMF mutation, whereas the orange sort of needle point there is the width of the wild-type allele. So there's definitely a much larger haplotype that's associated, and it's on the tail of the bell curve. And in fact, one can even calculate a selection coefficient for this mutation. It turns out to be 0.077, which is higher than the selection coefficient for lactase persistence in Northern Europeans, which is 0.056. In lactase persistence, the ability to digest cow's milk as an adult is something that has been thought to be under selection in the Northern European population. So just to sum this up and maybe to think a little bit then about the human condition, we have this organism, Yersinia pestis, that leads to the phosphorylation of pyrin as a way of basically sabotaging the immune system. You have a disease, familial Mediterranean fever, in which that's blocked by virtue of the mutations in pyrin. But even for individuals who are carriers, who are just heterozygous carriers, they're protected as well. And so you have this Faustian bargain, if you will, between us humans and plague, whereby we get some resistance to plague in a larger proportion of the population in exchange for individuals developing a lethal illness, a potentially lethal illness that's much less common because of just the fact that it's a recessively inherited disease. So anyway, so that's one thing to think about. Now another thing that you can think about, one can think about, I must say I think about every once in a while, is growing old. And so growing old and the risk of autoinflammation, that's the next topic here, having to do with the human condition. Of course, you know, it's a lot better to grow old than not to grow old. So you know, you have to think of it in that way too. And this is the work of David Beck, who was an excellent postdoc in my lab, who's now a junior faculty member at NYU. And then Peter Grayson, who's a tenure-track investigator in NIAMS, Marcy Ferrata, who's a scholar in NIAMS, and Amanda Umbrello, who's a staff clinician in my group. And so basically, these were the young and impatient. So they felt that we were not going fast enough in terms of finding disease genes. and that one really should take some sort of other approach than the sort of stodgy old approach of phenotype first. So phenotype first is where you group together a bunch of patients that have some phenotype in common, and then you try to find a gene that explains that phenotype. So that's tried and true way of doing it. But the genotype first approach instead is that you come up with a list of genes that you're interested in. And in David's case, he was interested in all of the genes that have anything to do with regulating ubiquitination. Now, for those of you who are not familiar, ubiquitination is just this post-translational modification of proteins that's really important in terms of determining what happens to the protein, whether it stays around or gets degraded in the proteasome or whatever. And we had actually discovered a couple of autoinflammatory diseases caused by mutations in ubiquitination genes, and so David thought that was a good place to start. And then he used that list of 841 genes to interrogate 1,477 exomes or genomes from our cohort and 1,083 exomes or genomes from the undiagnosed diseases cohort. He had various filtering criteria where he was looking for genes that are very intolerant for variation. That's the PLI greater than 0.9. Variants that aren't present in this big database of variants and that are shared amongst cases. And he found, after doing this Herculean feat of bioinformatics, that he found 3 middle-aged men who seemed to be heterozygous for a variant at position 41, at residue 41 of this UBA1 gene. Now, that seemed pretty good on the face of it, except that UBA1 is like the gene that encodes the protein that's involved in initiating all ubiquitination. And so it would be like, well, how can you live, if you have, serious mutations in that gene? So that was one problem. But then the real problem came up, and that is that UBA1 is encoded on the X chromosome. And these patients are men who apparently are heterozygous for variants of a gene on the X chromosome. So how can that be? Men only have usually one X chromosome. So is it a sequencing error? Well, David checked. It's not a sequencing error. I wouldn't be telling you about this if it was a sequencing error. Was it aneuploidy, an extra X chromosome? We checked. It wasn't that either. So then the third possibility is, could it be a somatic mutation where the DNA was derived from peripheral blood leukocytes, and rather than all of the leukocytes having both the wild type and the mutant form, that some of the leukocytes would have the wild type form and some of the leukocytes would have the mutant form. That was the idea. And so David proceeded to test that hypothesis, and sure enough, he was right. And so if you look at the neutrophils and monocytes, the myeloid cells, in fact, they have the mutation at this residue, whereas lymphocytes, T cells, and B cells do not. So that was really neat and interesting, and so we found evidence of a somatic mutation in these middle-aged men with unexplained inflammation. But then comes the fact stranger than fiction. And that is that we went to see Kathy Calvo in the Hematopathology suite. And because these patients had a hematologic condition, they all had bone marrows. And so we looked at the bone marrows. And sure enough, each of the patients had these funny-looking vacuoles in the myeloid and erythroid precursor cells. And Kathy said, well, she'd seen these before somewhere, but she couldn't remember where. And then we came back a few days later. Very ceremoniously, she presented me with 2 reports of patients that had these vacuoles. And she said, Dan, here they are. These are your patients from 8 years ago. And sure enough, they were patients of mine that had had a bone marrow from 8 years before. And we looked, and sure enough, they had somatic mutations, myeloid-restricted somatic mutations as well. They were, middle-aged men that appeared to be heterozygous for UBA1. and one of them had relapsing polychondritis. And this is just the wonderful thing about being in the intramural program of the NIH. So you find these things and you talk to somebody and you get another clue, and then that sends you to somebody else. And before you know it, we actually, with Peter Grayson and Marcy Ferrata and others, we had a cohort of 25 patients that all of them were men with unexplained or somehow explained fevers and inflammation that had various diagnoses, had carried various diagnoses: relapsing polychondritis, Sweet syndrome, myelodysplastic syndrome, polyarteritis nodosa, giant cell arteritis. But they all had mutations in this UBA1 gene. And so, being aficionados of naming things, we thought, well, this is a naming opportunity. We should call this something that really conveys what's going on with these patients. Let's call it Vaxis. And so Vaxis stands for vacuoles, because these patients, most of them have vacuoles, E1 ubiquitin activating enzyme, X-linked autoinflammatory somatic syndrome. And so that's what it's called. And David actually published this in the celebratory New Year's Eve edition of the New England Journal of Medicine at the end of 2020. So that was really neat. But then, what has actually happened is that it turns out that Vaxis is a lot more common than what we had ever thought. And so David, in collaboration with Doug Stewart in the Cancer Institute and people at the Geisinger Clinic in eastern Pennsylvania, did a population-based study looking at the frequency of Vaxis in their clinic population. And it turned out that there were 12 individuals who had been genome sequenced who were positive out of 163,000. So that works out to be, if you look in the men, 1 in 4,000 men over the age of 50, which is actually quite a lot. An estimated population prevalence of about 74 per million across age groups. And it's more common than a lot of the diagnoses that, were the diagnoses that these people had before, and it extended the clinical phenotype to a number of other things as well. So in any case, we're getting close on time. So fortunately, we're getting close to the end, though, which is unusual for me. So in any case, the last little vignette is a story about common genetic variants in the spectrum of disease. So this little segment on this slide, we're looking at images of patients with Behçet's disease. And for those of you who are not familiar, Behçet's disease is another disease that's seen in the Middle East. It's actually seen across Asia, even to China and Japan. And it's characterized by the triad of painful oral ulcerations, ocular inflammation, and genital ulcerations. And some patients develop pathergy, which is where they develop pustules in the skin if you prick the skin with a sterile needle. So in any case, we've been interested in Behçet's disease for the last 15 years. We've found at least one monogenic condition that sort of is like Behçet's disease called haploinsufficiency of A20. But we've also done a number of GWAS studies and have identified, besides HLA-B51, which is well known, 17 other genes that are involved in susceptibility to Behçet's disease. And this was done with a very careful study with Ahmet Gül in Istanbul, where he carefully phenotyped 1,000 patients and 1,000 controls. It was really a huge job. And then, after we had completed that work, the direct-to-consumer company 23andMe came out with a genome-wide association study of canker sores. And all they did was, in a couple 100,000 people, they just asked them, do you get frequent canker sores, yes or no? And then they did a GWAS on that information, and it turned out that a lot of the same genes that we had found in Behçet's disease are also associated with just common canker sores. And some of the ones that are particularly interesting, interleukin-10, interleukin-12 alpha, STAT4, a non-coding variant of STAT4, and CCR3. And then what happened is that Kalpana a fellow in my lab who's now an ACI and NIID, was studying a condition in children called Fapa. syndrome, which stands for periodic fevers with aphthous stomatitis, pharyngitis, and cervical adenitis. So these kids get sores in their mouth, as well as sore throats and swelling of the lymph glands in the neck. So it's the most common periodic fever syndrome in children. It's not monogenic. It's a genetically complex disease. Most kids grow out of it. And because it's sort of in between Bertels disease and common canker sores, Kalpana looked to see if these same genes are associated with susceptibility to Fapa, and yes, they are. So you have this spectrum of diseases that on the one end is severe with Bertels disease, on the other end, recurrent aphthous ulcers, and then Fapa in the middle, where it seems like there's a cassette of common variants that somehow are predisposing to certain immune or inflammatory phenotypes. So this is it, the 30,000-foot view. Monogenic autoinflammatory diseases have taught us a great deal about how innate immunity works and its intrinsic connection to basic cell biology. Targeted therapies have been transformative while validating our understanding of the underlying disease. Variants in the innate immune system may protect against pathogens but predispose to autoinflammation. Somatic mutations account for at least some adult-onset autoinflammatory diseases. We think that there's probably a bunch more to be discovered. And cassettes of relatively frequent genetic variants confer risk for some common autoinflammatory conditions or phenotypes. So in any case, at this point I'm wrapping up. I do want to thank my colleagues in the lab. Pictured here are just current colleagues in the lab, and in particular two extraordinarily productive and competent staff scientists, Ivona Aksentievich and Jay Che, but certainly a number of others to be mentioned, colleagues in the clinic. And I do want to emphasize that this really is very much a partnership, a collaboration between the lab and the clinic. Staff clinicians, Amanda Umbrello and Debbie Stone, shown here in the upper left, but a number of other individuals as well. Quite a few collaborators, and again, these are just some of the more recent ones. And then, of course, our patients. This just happens to be the meeting, the first international meeting of patients with, well, on DADA 2, which was back in 2016. And, you know, this work just would not be possible without patients really playing a major role. And then a number of people that have contributed in various ways to my career. Just very briefly, Bob Rich, who was my graduate school advisor, and it's half a century ago now, introduced me to all of the fun and excitement of immunology, such as it was in the 1970s. The late Fred Steinberg, who hired me as a clinical fellow at the NIH back in 1985. Paul Plotz, who was really a role model in terms of being a physician scientist. The late Henry Metzger, who was my scientific director when I started as a tenure tracker. And I will never forget the words that he said when I started as a tenure tracker. He said, Dan, here's some resources. Just go and have some fun. And be sure that you'll have some data ready in 18 months when you present to the Board of Scientific Counselors. You know, so that was Henry. And certainly I appreciate the latitude that he extended. Mordecai Pross, my collaborator in Israel. Mike Dean of the Cancer Institute, who really taught our group how to do genetics and genomics at scale. That was before there was a Genome Institute. Paul Liu, who's been a scientific and administrative collaborator for many, many years. The late Steve Katz, who was the director of NIAMS and certainly a great supporter of my work during the time that he was the institute director. Eric Green for being a wonderful boss and scientific and personal collaborator as well. And then Michael Gottesman, sitting here in the front row. And I just do want to say that, you know, Michael, for 3 decades, created a culture here in the intramural program. We talk about, you know, having a scientific community in which we value long-term, high-risk, high-reward projects. And certainly where collegiality and collaboration are the watchwords of what we do. He's the man that really inculcated that into our community, and I think we owe him, all of us owe him an incredible debt of gratitude for that. And then finally, here, oh, it's a blank screen, I'm looking ahead. My wife, this is our wedding day back in 1985 when we were just about to set out on our big adventure here in Bethesda. And then about a year ago when we were in Sweden when I got the Crawford Prize. So, you know, really she's been a source of support and strength and just a best friend over all of that period of time. And then finally, the last slide here, the NIH Clinical Center, which really made all of this possible. 3 institutes that have played a major role in my work, the NIAMS that I started in, NHGRI where I am now, and also the NIAID, which has provided great collaborations on a number of clinical topics, especially over the last couple of years. So with that, I will call it to a close. I see that we have about 3.5 minutes for questions. So anyway, I'll try to answer your questions.
You can stay a little late. How do you explain the late onset of disease in these patients if the mutation has been there for the individual's entire life? Is there a missing variable that is not accounted for?
Yeah, well, of course, for those diseases where there are germline mutations, that is a question that comes up. Certainly for a lot of these, the diseases that I was talking about that are the monogenic diseases, they present by adulthood anyway, and so it's not as if people are going for a long, long time. But it's still a point well taken that we do not understand all of the mechanisms of these diseases. You know, we've tried making mouse models. We tried, for example, with familial Mediterranean fever, to make mouse models of FMF, which we did make, it's true, and they have continuous inflammation rather than episodic inflammation, so then that, you know, precluded our being able to look for provocative factors. And the interesting thing also was that we did 3 different mutations of different severity in humans. They're also of different severity in mice, but the exact opposite. So the most severe in the human is the least severe in the mouse, which I guess is just another demonstration that we are not mice. So anyway.
Yeah, good to clarify that. Another one, Dr. Kastner, always a fascinating talk, exclamation point. Could you share thoughts on secondary, not germline, autoinflammatory features due to uncontrolled long-term autoimmune disease? More specifically, can epigenetic changes from chronic inflammation make it look like the new onset of inflammatory disease? Any links to mitochondrial dysfunction in that sense?
Those are all excellent ideas. We have not actually tested those hypotheses, but certainly the questioner is correct that there is very much an interaction between the adaptive and the innate immune system. And so one would expect that if someone has germline mutations or even susceptibility variants from one side or the other, that one could imagine that over time that one could get secondary effects that would bleed over into the other domain of the immune system.
Okay, and I'll wrap up with 3 points. CME code, I think, was 44460. I hope so. There will be a reception outside for people here, of course, and I'm pretty sure the history office would love a copy of that handwritten letter someday, should you want to bequeath it to them. Okay.
All right. Well, thank you.
Thank you.